Hello everyone, this is Richard from Modern Healthspan. Today we will go through a study of a promising new use of a molecule that activates SIRT1 in the skin to help reduce senescent cells. The compound is already in use as an anti-influenza drug, and after the research, they have already filed a patent for it as a skin treatment. First, a disclaimer that in this video we are sharing a study that we found interesting. It is not a recommendation or medical advice. Here is the paper. Shikimic acid protects skin cells from UV-induced senescence through the activation of SIRT1. Let's have a look at the paper in more detail. UV radiation is one of the main contributors to skin aging by promoting the accumulation of senescent cells, which in turn secrete chemicals which are pro-inflammatory and degrade the tissues, leading to skin aging. The SIRT1s play an anti-senescent role, and their activation suggests a promising approach for preventing this UV-induced senescence. To address this, first the authors screened a number of compounds and identified shikimic acid as their best candidate. The study uncovers a shikimic acid SIRT1 signaling pathway that prevents senescence but also reinforces the role of SIRT1s as a key regulator of the proteins in the cell. So what is shikimic acid? Here is the structure of the compound. Shikimic acid, or shikimate, is a metabolite common in plants and microorganisms. Clinically, it is already used as the active ingredient in Tamiflu, a medicine to prevent H1N1 swine flu. In the study, the first step that the researchers did was to screen a number of known or suspected SIRT1 activators to determine their efficacy. In the screen, they had two tests. The first was to look for SIRT1 activity, which they did by looking at specific changes on the epigenome that SIRT1s perform. These two columns are markers which are removed by SIRT1, so the authors wanted to see both of them go down, which is what we see for shikimic acid. And the second was to look for markers of senescence after exposing the cell culture to UV light. In this case, the first of these is beta-gal, a marker of senescence, and the second is the expression of hyaluronic acid synthase 2, or Has2. They wanted the first of these to go down and the second to go up. And again, this is what we see for shikimic acid. Having found the molecule that was most effective, they wanted to identify its mechanism of operation. The first test they did was to irradiate some human dermal fibroblasts, or HDFs, with UV and look for the same markers of senescence that they used in the previous test. In this case, they had a control and other samples with various concentrations of SA. We can see that the amount of beta-gal is reduced with the addition of the shikimic acid. However, the addition of EX527, a SIRT1 inhibitor, undoes this effect, showing that SA is working through SIRT1. They think that the mechanism of action is that UV light causes an increase in misfolded proteins, which in turn activates the unfolded protein response, or UPR. The UPR's role is to remove these proteins. However, when overactivated, it can in turn overactivate autophagy and then push the cell towards senescence. Shikimic acid upregulates a protein called BIP involved in the folding process, reducing the number of misfolded proteins and so the need for UPR, which is then downregulated. It seems to do this through SIRT1, as again, when EX527 was used, the behavior was reversed. The exact path through which shikimic acid activates SIRT1 was not found, and it does appear to be indirect. The blue dotted pathways between shikimic acid, SIRT1 and senescence indicate that the authors postulated that there may be other mechanisms, but these were not identified. Here is their conclusion. SA has low toxicity and appears a good candidate for treatment of skin senescence and aging induced by UV irradiation. They do not mention it in the paper, but if the compound can improve the protein quality control in the cell, it may have wider applicability beyond just UV-induced damage. In the conflict of interest section, as mentioned earlier, we see that two of the authors work for a company that has applied for a patent to use shikimic acid for skin treatment, and it is already being used for an approved treatment, Tamiflu. So this looks promising, with the treatment already a couple of steps on the way to being commercialized. I hope that you found the video informative. Please do hit the thumbs up button, subscribe to our channel, and hit the bell button for any new video release notifications. Thank you so much for your kind support. I wish you all well and will speak to you again soon.